This is the third video in the muscle tissue series. Recapping from the previous video, you should feel pretty comfortable identifying the parts of the thick and the thin filaments. Remember that the thick filament is made up of myosin, and that flexible hinge region is going to be really important in this video. And then the thin filament is made up of actin, troponin, trypomyosin, and you should feel comfortable with each of their roles. If you're not, then please go back and make sure you have a good grip on the content in that previous video before you continue on with this one. Okay, because in this video I'm going to just use these terms, troponin, trypomyosin, myosin head binding sites. On actin, I'm going to use those just very, very comfortably, and so you should also feel comfortable with those terms. Okay, our learning objectives is number one, what is the role of calcium? You know, up till now we keep talking about calcium. We've seen that over and over and over. You know, that calcium was spilled out over here, and calcium ended up binding to troponin. And so you need to understand what calcium is doing for us. What are the two configurations of myosin? I don't expect that sentence alone to make a lot of sense, but that's going to be a major topic we cover. What is a cross bridge? And then finally, what is the role of ATP? And there's two things that happen with ATP. When ATP itself shows up, and number two, when we actually burn or hydrolyze ATP. So we have to know what those are. So the first thing is, let's actually go into the two configurations of myosin. So what does configuration mean? Configuration is just a fancy word to say shape or state of myosin. So you already know what myosin looks like. So you have this image in mind where you got the tail, that flexible hinge region, and the head. So what I'm going to say is that this myosin molecule can exist in one of two states, one of two configurations. And we're going to describe those as a high energy configuration and low energy configuration. So those terms, if you had to really sit and think about it, you'll find that they're not very far removed from your intuition. If you look at this picture here, this is a rubber band just sitting there. You can ignore this box, unstretched rubber molecules and stretched rubber molecules. You can ignore that. But if you look at this, this is a rubber band in an unstretched state, and now I've taken the two ends of it and I've started to stretch out the rubber band. So again, this is my unstretched state and this is my stretched out state. So I've grabbed and I've tugged and pulled on the two ends. And so if I were to say to you, which of these two rubber bands, this one here up top or this one below, which one of those has more energy in it? Which one has stored energy? All of you would say that this one, the stretched rubber band, does. You know, an unstretched rubber band is not harmful or dangerous at all, but that stretched rubber band, you've all been around people who have like pulled rubber bands out and you're afraid that they're going to actually let it go and it's going to smack you in the face or something. There's a lot of stored energy in there, and then when that stored energy is transferred to your face, it hurts. And so it's probably not far from your imagination that if I were to label this as the low energy configuration of the rubber band and this the high energy configuration of the rubber band. And so again, configuration is just a fancy way to say the shape or the status of that rubber band. So again, low energy, not very active, high energy. Okay, lots of stored energy in there. So we're going to do the same thing. Now, instead of a rubber band, I want you to picture the myosin molecule itself. So again, you already have this image in mind. There's the tail and the head. And what I've done is I've drawn out myosin for you. So these are the two configurations of myosin. This is the high energy configuration where there's the myosin tail. This is that flexible hinge region. And then there is the head. This is in a fully stretched out state. So myosin is in a stretched out state here. If you look at this one here, this is your low energy configuration, and this is the released rubber band. This is a rubber band just kind of completely released. So I'd put that little hashed mark arrow here showing you that this is the way the rubber band wants to go. In this case, it's the myosin molecule. This myosin head really wants to go out in this direction, because there's stored energy here, and if you were to let go of one end of the rubber band, so to speak, we let go of this end, then that hinge is right over here at the neck, and then we have, at rest, the low energy configuration. So again, lots of stored energy like the stretched rubber band, and a low energy configuration. Lots of stored energy and a stretched rubber band, and a low energy configuration. So that's how we end up thinking about myosin. So when the muscle itself contracts, we really depend a lot upon this activity of the myosin head, the myosin molecule going from high energy to low energy, and then to high energy to low energy. So if we look at the way that myosin molecule is actually set up in the context of everything, imagine all these myosin heads, again, reaching out and grabbing onto that thin filament. And when they grab onto the thin filament, they are in a stretched out high energy state. These myosin molecules is the same thing. Their tails are over here and the heads are over here. They're in a stretched out state. Now, when they go into a low energy state, what are they going to do? They're going to go ahead and they're going to start looking like like this. Or we create a mirror image of this thing and it looks something like that. So what you can imagine then is if I put in an M line, let's say I took a line and I made this the M line right over here. And then what I did is I took a bunch of these. In fact, let me do that on another slide. So if we look at it, it might look something like this. Here's my high energy configuration. And again, if I slap together a bunch of these guys, there's a bunch of myosin molecules in a high energy configuration this way, and there's more this way. So what that should be doing, that should be paralleling this picture over here, okay? High energy configuration. And what ends up happening is that once they grab onto the thin filament, which I haven't shown you in this particular slide, but assume there's a thin filament here. Actually, if we redraw it, it might look something like this. And so we have all your myosin heads stretching out, grabbing onto the thin filament, and these are now in the high energy configuration. And when they're allowed to release their energy and go into that low energy configuration over here with the mirror image over here to the low energy configuration, you see what's going to happen is that as they grab onto the actin filament, that thing is going to pull inward. Again, of course, attached to the Z discs and such. So this is just a quickly sketched out graphic within PowerPoint. But let's show you what it actually looks like when a better artist draws this thing. So if you look here, this is a cycle, and this cycle is going to show you a muscle that's going to continue to contract. Imagine taking a plate, a heavy plate off the table and holding it with your elbow flexed at 90 degrees and just holding that plate there while you're talking to somebody, having a conversation. So in that state, your muscle is continuing to contract. If you doubt that, and again, just grab like a 10-pound weight or 5-pound weight and flex it up, bring that weight not quite to your shoulder, but halfway through, and just hold that weight there. Your muscle continues to contract. And that's what we're seeing here, is we're seeing a continuous contraction cycle. So we're going to jump in somewhere, and I'm going to walk this through 3 times. This is myosin. In this state, myosin actually is in a high energy configuration, and there's ADP and P attached to the myosin head. Where did it come from? We'll get to where it came from. So myosin head is in a high energy configuration. You see that tropomyosin has already been dragged away. So myosin head wants to bind to the actin filament, and it binds. From there, again, my rubber band is in a stretched out state. I'm right now in this state, the stretched out state. I am in my high energy configuration. I am in my stretched out high energy configuration. So what do I do from there is that, well, I let go. And when I let go, what ends up happening, that myosin goes from high energy configuration into a low energy configuration. You see it go through this stage and then this stage. So what ends up happening is as that rubber band goes back to the low energy unstretched state, you end up releasing ADP and P. The inorganic phosphate is the P. So that comes along. From there, a new ATP will show up. And honestly, if I had my dream here, not my dream, but if I had my own wishes, then there would be one more image stuck right in here. And what that image would show you, it would show you this ATP molecule floats along and comes and attaches to the myosin head. So after ADP and P are released, my myosin went from high to low energy configuration. This is low energy. And then a new ATP molecule comes along and it binds to the myosin head. When the ATP binds to the myosin head will then detach from the actin filament. So read this right over here. As new ATP attaches to the myosin head, the link between myosin and actin weakens and the cross bridge detaches. That means the myosin head detaches. So from there, what do? We're going to burn ATP. We're going to spend that ATP. We're going to release that energy. And that energy is used to take the rubber band from an unstretched state, low energy, and it's going to bring that back out to a high energy state. And that's the direction of this arrow over here. So when I hydrolyze ATP, what I'm really doing is I'm taking these myosins and I'm bringing them back to this. When I bring them back to this state, they find another myosin head binding site. And then from there, I'm going to release ADP and P. Where did that come from? Oh, it came from the ATP that I just burned. And from there, I go back into my low energy configuration. A new ATP molecule will show up, binds to the myosin head, in which case the myosin heads will now detach from actin. I burn ATP, which makes me go back to a stretched out state over here, in which case I find another myosin head binding site on actin, and then I continue to do that over and over on this slide again. And so I brought myself back to high energy configuration by burning ATP and stretching me back out. From here, I find another myosin head binding site. I release ADP and P, and I go through this motion going from high energy to low energy. Now, that action of going from a high energy to a low energy configuration is called the power stroke, and that's this thing right over here that I'm highlighting, or that I'm circling. This is called your power stroke. So my myosin head undergoes the power stroke. From there, a new ATP shows up, binds to the myosin head detaches, and I stretch back out. From there, here I go. I burn ATP, I stretch back out, and I find another myosin head binding site on actin, and I continue to do that thing over and over and over again. Again, alternating between high and low energy states of my myosin. This is a really nice video showing you on YouTube, and I will definitely link this up within our Canvas site. But let me just play a short clip of it. It starts at 51 seconds, and I'm just going to play a short clip. What we see already is the actin, the troponin, and these little snake-like things are tropomyosin, and here's my myosin molecule. So there's my myosin. Calcium comes and binds to troponin. Troponin changes its shape, and when troponin changes its shape, it reveals the myosin head binding sites on the actin filament. From there, now you're going to see that myosin head wants to bind. That's its default status. So the myosin head itself will want to bind, and because it wants to bind, it's going to go ahead and find that section of the myosin head binding site on the actin filament. There we go. And I release the phosphate en route. Here, now I'm in a stretched out state, right? I started off in a stretched out state, and now I'm going to my unstretched state. My rubber band is going back to its unstretched state now. You're about to see a new ATP molecule show up because myosin has got to detach. If you want to detach the myosin head so you can stretch back out to do it all over again. So there's ATP. ATP showing up detaches the myosin head. When ATP gets burned, what we're going to see now is I'm going to go from a low energy configuration back to a high energy configuration here. That stretches out the myosin head. And burning ATP then leaves me with ADP and P. From there, again, as long as the binding sites are available on the actin filament, I'm going to continue to do my thing. So that cycle that we just saw is going to repeat itself over and over and over. So again, I'll put a link to this up on, actually I'll go ahead and embed this within our Canvas site, but go ahead and watch this through and narrate it through. I would strongly encourage you to narrate as you watch it. So a couple of major assumptions that happen here that I want you to know. The first assumption is that myosin wants to bind to actin, and as long as those sites are unveiled, myosin will bind. So as long as the binding sites, the myosin head binding sites on actin, remember those darkened pits, those darkened black portions of that bead of pearls right over here? See these darkened areas? And so, well, what do I mean veil and unveiled? It means, remember, tropomyosin covers them up. But when tropomyosin gets out of the way, they're now unveiled. So as long as they're unveiled, myosin is going to hunt for it and it's going to find it. That's our first assumption. The second is this. If the binding sites on actin are covered up, then myosin cannot bind and the muscle is going to be at rest. The muscle will not contract. And number three is that you'll never run out of ATP. And what I mean by that is, here's always the question. The question is, how long does this cycle continue? You'll see that this is in a cycle. How long does that cycle continue? And your answer, on the one hand, you might say, well, as long as ATP is available. And I'd say, well, in principle, you're correct, but you'll never run out of ATP. The moment you run out of ATP is the moment you die. And so the skeletal muscles are chock full of mitochondria. They're very oxygen-hungry molecules. And so, sorry, not oxygen-hungry molecules. They're very oxygen-hungry cells because they have so many mitochondria in them. So you really will never run out of ATP. So how long does this cycle continue? Well, the answer is this cycle continues as long as myosin head binding sites are available. So the next question is, well, how long are myosin head binding sites available? And you say, as long as troponin is not covering them up. Well, how long is troponin not going to be covering them up? As long as calcium is bound to troponin. Well, how long is calcium bound to troponin? Well, as long as calcium is available in the sarcoplasm. Well, how long is sarcoplasmin available with the calcium? As long as I keep stimulating the sarcoplasmic reticulum. How much am I going to keep? How long am I going to keep stimulating the sarcoplasmic reticulum? As long as I keep generating action potentials. In other words, as long as the nerve fires. As long as your nervous system wants to, the muscle will continue to contract. Okay, and so this will continue as long as calcium is available. Now, one thing I do need to tell you: this sarcoplasmic reticulum, there's a pump on here, which you're not visualizing here, and that pump is constantly sucking back calcium. So this calcium spills out, but as soon as it spills out, I mean, just a brief moment later, it's being vacuumed right up. That vacuum is always, always on. And so I want you to imagine that that vacuum operates at sucking up 10 calcium ions per second. Well, if I spill out this many, it's going to take me a couple of seconds to vacuum all these up. So calcium will diffuse off to wherever troponin is within my sarcoplasm in this area. But what ends up happening is that unless I continue to stimulate that sarcoplasmic reticulum with consistent, constant barrages of action potentials, eventually the muscle is going to relax because my vacuum will eventually suck up all this calcium. Let me say it differently. Your muscle will continue to contract as long as the nerve continues to stimulate the muscle. If the nerve stops stimulating the muscle, then you no longer send action potentials, you no longer release calcium from the sarcoplasmic reticulum, and then within 2 seconds or whatever it might be, all those calciums that were available in the sarcoplasm again is the cytoplasm of the cell, those calciums are now all sucked back up into the sarcoplasmic reticulum. All right? So, make sure you understand this section. Everything we've done here, if we were to rename this really is, this video really is the sliding filament model. And so you can Google sliding filament model. There's a ton of YouTube videos that show different animations that look at this. The one I showed you was just one that I found. I like it. But this idea of high to low energy configuration and this whole cycle, you should feel very comfortable talking this through. Okay, let's see. You know the role of calcium. Calcium binds to troponin to trigger a change in shape of troponin, which then drags away tropomyosin from the myosin head binding sites. The two configurations are high and low energy. What is a cross bridge? A cross bridge is simply when myosin heads bind to actin. And what is the role of ATP? Well, ATP, when it binds to myosin, it will detach myosin from actin. And when ATP is hydrolyzed, that will make the myosin molecule go from low energy to high energy configuration. And that will be the end of this video.